Hi guys, this is Kumar. In today's lesson of genetics, we are going to learn how to make the Punnett square of a monohybrid cross. So if we start today's lesson, you need to know some of the vocabulary words like dominant, recessive, traits, genes, purebred, crossbred. If you know these words, you're ready for today's lesson. If you're not, go back to my first lesson of Introduction to Genetics and watch it, and then you are ready for this. But if you know all these words, you're ready for today's lesson, and let's start that how to make a Punnett square of a monohybrid cross. Before you learn today's lesson, you need to know two of the vocabulary word. One is genotype and phenotype. I would like to explain these two words first so that you can understand how to make a genotype and what's the importance of making a Punnett square. Genotype is nothing but a combination of genes an organism has, and you can also say that the actual makeup, we call that as a genotype. And what this genotype represent is in some of the combination of alphabets. I'm going to discuss later what these alphabet represent and how you can have for some different combinations. What is phenotype? Phenotype is a physical appearance resulting from these genetic makeup. We call that as a genotype. So physical appearance resulting from gene makeup, we call that as a genotype. Looking at this picture, if genotype is the combination of all these genes human beings have, these genotype are going to code for the phenotype, which are the traits of the organism. There are all kinds of variation if we are talking about phenotype and genotype, and there are so many ways the gene can express themselves. It's often that one genes are a combination of genes that give an organism a particular trait. Since there are so many possibilities when the genes of two parents combine, it's helpful to use a tool called Punnett square. What is a Punnett square? Punnett square is nothing but a graphical way of determining all the possible genetic outcome when the cross is performed. When I'm talking about the cross, that means when you're crossing a male and a female, what is going to be the outcome? Outcome is we are talking about the offspring. Punnett square is also a probability box that shows the odds of each possible traits happening in the next generation or the offsprings. As you know, when traits are expressed by an organism, they result from two copies of genes. One copy is coming from your mother and one is coming from your father. And when we are talking about genes can be of two types. One is dominant and the one is recessive. If the recessive is present, it will be masked by the dominant gene, and always the dominant is going to be the boss, which is going to decide what the trait the offspring are going to have. You can say the dominant is the boss and decides for the traits an organism or the offspring is going to have. The dominant is represented by the capital letters. If we are talking about the eye color and we say the black eye color is dominant, then for this black dominant eye color, you're going to use a capital alphabet like B. And if you're talking about a recessive, which is going to be represented by a lowercase, and if you're talking about the eye color, blue color is recessive, then it will be represented by a lowercase b. Always remember, the dominant is going to be the boss and decide for the trait in the offsprings. There can be three possible outcome of the genes if you're talking about. One is homozygous dominant. The homozygous dominant is when the both genes are the same and dominant. Another option can be heterozygous. So heterozygous is a dominant gene when there is a dominant gene present and a recessive gene present. You know that dominant was represented by capital, this is B, and this is a lowercase b. This is a case of heterozygous. Hetero means different, and there are different two genes present, which were the option was the dominant and the recessive. The last option present is the homozygous recessive, which will be having two homozygous, that is the two recessive genes present. And in this case, since there is no dominant gene present, these two recessive are going to express their traits. If we were talking about the eye color, then if an organism is going to have these homozygous recessive, they are going to be having the blue color eye. Let's have a recap. If we are talking about homozygous, there were two possibilities. One was the homozygous dominant. If you are talking about homozygous dominant, it's going to be two capitals. And if you are talking about homozygous recessive, they're going to be two lowercase. Another option is going to be heterozygous. When we are talking about heterozygous, we are saying that there is going to be a presence of one dominant and one recessive. This was the three possible outcome of the genetic makeup, genes which can be passed on or to the offsprings. If you're talking about genes, and if you're talking about the phenotype and genotype, when the offsprings are produced, the parents can pass either of the genes. This is why a Punnett square is a helpful tool in predicting the phenotypic and the genotypic ratio. When you're going to make a Punnett square, you have to make 4 square boxes. And always remember that on each side, place each parent's genes on the outside of the box. If, let's take example, if you're crossing a homozygous dominant with a heterozygous, they're going to be, you have to place, this is coming from the mother, this is coming from the father, you have to place each parent's genes on the other side of the box. So it doesn't matter where you're going to put that, but you have to put like this. And this is from the mother, this is going to be father, that was the heterozygous. To predict the offspring, you have to write the genotype in the boxes. So this is going to be 2 capitals, 1 capital with a lowercase, which is going to be heterozygous. Again, there are going to be 2 homozygous dominant, and this is going to be another heterozygous. Let's take an example to understand how to make a Punnett square and identify the probability of a genotype and a phenotype. As you already know that Punnett square is used to predict the possible gene makeup of an offspring. In this case, we are going to take an example of black, which is a dominant fur in mice, and it's going to be represented by a capital B. And a white fur, which is a recessive, is going to be in a lowercase b. In this case, let's cross a heterozygous male and a homozygous recessive. So the genes for the male are going to be heterozygous, so they're going to be a dominant and 1 recessive. For female, it's going to be both recessive since it's a homozygous recessive. To make a Punnett square, remember to place each parent's genes on the outside of the box. As the genes for male were heterozygous and female was homozygous recessive, place these genes of male gamete, that is 1 gene in the sperm, it's going to be a big B and a lower b. and 2 lowercase b's for the white fur. Let's see what are going to be the outcome in the form of offspring. They're going to be a 1 heterozygous, another heterozygous, since it's coming from this and this. It's going to represent a heterozygous. Another one is going to be B and B, which is a homozygous recessive, and another homozygous recessive. So let's see what is going to be the genotypic ratio. So in this genotypic ratio is going to be, we have 2 heterozygous and 2 homozygous recessive. It's going to be 50% of heterozygous and 50% homozygous recessive. And these genotypic ratio are going to represent the phenotypic ratio. So there are 2 heterozygous. Since there is a presence of a dominant, it's going to be black, and there is a lack of dominant here, so 50% mice are going to be having white fur. Genotypic ratio was 50% B heterozygous, 50% homozygous recessive, and phenotypic ratio was 50% black and 50% white fur. Let's take another example. We are going to cross 2 hybrid mice. When I'm talking about hybrid, that means it's going to be heterozygous. So a cross between heterozygous and a heterozygous, it's going to give these 2 genes, and in the boxes you're going to have a big B and a big B, which is a homozygous dominant, and another one is a heterozygous, another heterozygous, and a homozygous recessive. Let's predict the genotypic ratio. You can see here there are 2 heterozygous, 1 homozygous dominant, and 1 homozygous recessive. Genotypic ratio is going to be 1 to 1, and this is going to predict the phenotype. Since there is presence of 1 dominant, you can say there is going to be black fur, another black fur, but there was lack of a black genes. This is going to be white. Phenotypic ratio is going to be 3 black and 1 white. So this was a cross between 2 hybrid mice. When we are talking about hybrid, that means it's a heterozygous. If you want to have more practices, you have to go for some doing different combination of crosses and make some practice your own Punnett square, and that's how you're going to learn to predict the genotypic and the phenotypic ratio. So that was all for today. See you in the next video, in which I'm going to discuss the dihybrids. That is, I'm going to take 2 traits and how they move or how they predict the genotypic and phenotypic ratio. Thank you.